Hey all, I'm Shanjitha Shanmugasundaram from Biotech Vibes with Shanji. This channel is my new initiative during this lockdown. In this channel, I have planned to explain precisely about biotech related topics, and for today's video, we are going to discuss about gene therapy in cancer biology. For queries, suggestions, and doubts, please don't hesitate to contact me through the given mail ID, and I will be providing this mail ID in the description box too. Gene therapy. The broad field of gene therapy promises the number of innovative treatments that are likely to become important in preventing death from cancer. In other words, we can simply define it as the type of experimental treatment in which foreign genetic material, DNA or RNA, is inserted into person's cell to prevent or fight against cancer. As we all know, there are two types of gene therapy. The first one is germline gene therapy, and the second one is somatic gene therapy. Germline gene therapy is nothing but the eggs and sperms are being altered by the introduction of functional genes. It is an inheritable one, which means it will be carried over generations. Somatic gene therapy, the therapeutic somatic gene therapies. The therapeutic genes are transferred into the somatic cells of the patient. Any modification is restricted with the individuals, which means it is not the inheritable after generation. Gene therapy in cancer biology deals with this type. There are three different approaches. First one is immunotherapy, second one is oncolytic virotherapy, and the third one is gene transfer. Immunotherapy in cancer. The concept of boosting the immune system to and destroy cancer cells has been the goal of cancer treatment for over 100 years. In simple words, immunotherapy can be defined as immunotherapy uses genetically modified cells and viral particles to stimulate the immune system to destroy cancer cells. We have three different modes. The first one is immunotherapy with altered cancer cells. The second one is the immunotherapy with gene in vivo. The third one is immunotherapy using altered immune cells. As in the first case, that is immunotherapy with altered cancer cell, the cancer cell and the genes are being incorporated and being injected to the host cell, and as the result the immune system is being activated and the tumor cell has been destructed. In the second case, the gene has been directly injected to the cancerous tissues and allowed to grow. And as the result, the immune system has been activated and obviously the tumor cells has been destructed. In the third mode, immunotherapy using altered immune cells, the immune system and the immune cells and the gene has been incorporated and allowed to grow. And it has been injected to the live cells and the immune system has been activated, which resulted in the tumor cell destruction. Recent trials in the second and third generation vaccines have shown an encouraging result with the wide range of cancer, including lung cancer, pancreatic cancer, prostate cancer, and malignant melanoma. As with any cancer monotherapy combination therapies, using vaccines may be more effective than the vaccine therapy alone, which means the combination of two therapies shows the effective result. Cancer vaccines have been presented only the modest immune response may find useful as an adjuvant therapy for using after surgery or chemotherapy to eliminate the remaining cancer cells. Gene in charge for immunotherapy. For prostate cancer, it is murine 1,3 galactosyl transferase. For pancreatic cancer, it is CEA and MUC1. For prostate cancer, it is GMCFF. For lymphoma cancer, it is GMCFF and CD4OL. For melanoma, it is IL2. For kidney cancer, it is CD80. The second type of approach is oncolytic agents. Another growing area of gene therapy treatment for cancer cells use oncolytic vectors of cancer destruction. In simple words, oncolytic virotherapy, which uses viral particle to replicate with cancer cells to cause cell death. The number of different viruses have been introduced for the purpose, including vaccinia adenovirus, herpes simplex virus, retrovirus, and Newcastle virus. Retrovirus. Retrovirus. The retrovirus is the type of RNA virus that inserts the copy of its genome into the DNA of host cells that invades, thus changing the genome of the cell. Once inside the cell cytoplasm, the virus uses its own reverse transcriptase enzyme to produce DNA from RNA genome. The reverse is the reverse of the usual pattern, thus retro. Retro is nothing but the backward pattern of the usual pattern. The new DNA is then incorporated into the host cell genome by integrase enzyme, at which point retroviral DNA is referred to as provirus. The host system then treats the viral DNA as a part of its genome, transcribing and translating the viral gene along with the cell's own genes. Producing the protein requires to assemble new copies of the viruses. The engineered oncolytic genes have been directly injected to the cancerous tissues and allowed to grow, and which obviously resulted in the cell lysis. Cell lysis is nothing but the cell death. Oncolytic vectors designated to infect cancer cells and induce death through propagation of virus, expression of cytotoxic protein, and cell lysis. It is an emerging treatment modality that shows great promises, and particularly in metastatic cancers. The third approach of gene therapy in cancer biology is gene transfer. Gene transfer is a new treatment modality that introduces new genes into the cancerous cells or the surrounding tissues and causes cell death or slow down the cell death, which is nothing but the new cancer cell is being introduced to the cancerous cell and causes the cell death, or else it slow downs the growth of the cancer cells. Gene with number of different functions have been proposed for the type of therapy, including suicidal genes, anti-angiogenesis genes, cellular status genes. This treatment technique is very flexible and a wide range genes and vectors are being used in the clinical trials with successful outcomes. In this method, the gene is being directly injected to the cancerous tissues and allowed to grow, and which directly resulted in the cancer cell lysis and destruction. Genes in charge for this gene therapy is for pancreatic cancer, it's Rexin and TNF alpha. For glioblastoma, it's HSBTK. For head and neck, it's P53. Talking about P53, it is also called as genome guardian. It acts to kill cancer cells, suppress cancer growth, and prevents the cell becoming cancerous. Genocide. It is the common drug that is being composed mainly of human P53 tumor suppressor genes and modifies adenovirus. Its functions are to triggering the activity of natural killer viruses and decrease the tumor resistance to radiotherapy and chemotherapy. Advantages of gene therapy with cancer. It has a potential to kill cancer. It gives chances for a life of someone who is born with mutated cancer gene. It can be used to eradicate disease from the future disease. Disadvantages of gene therapy with cancer. It is an expensive treatment. The gene therapy requires ongoing treatment to be effective. The genetic testing, screening, research in finding the availability of certain gene is very controversy. And we are into the end of the video. I hope it was an useful section for you all. Thank you. And if you like my work, please do subscribe my channel and press the bell icon. Thank you.